For decades, the clinical standard for photoprotection has relied entirely on a single mechanism, passive photon attenuation. You apply a chemical or physical barrier to the stratum corneum, and it absorbs or scatters incoming ultraviolet light. But passive barriers have an inherent limitation. No matter how thickly you apply them, they cannot block 100% of incoming solar radiation. This chart illustrates the reality of high-level protection. A properly applied SPF 50 formulation still permits exactly 2% of UV radiation to penetrate the skin. In a high UV tropical environment, that 2% represents a massive, sustained volume of absolute photons striking your cells. The biological consequences of this leakage are immediate. Bright sunlight penetrating that barrier causes adjacent thymine bases in your cellular DNA to fuse, forming undesirable chemical bonds at a rate of 50 to 100 bonds every single second. Beyond direct DNA damage, this residual UV exposure generates reactive oxygen species and free reactive iron, triggering a cascade of severe oxidative stress throughout the epidermal layers. Preventing long-term photoaging in extreme environments requires recognizing the limits of an impenetrable shield. We have to transition from a passive chemical barrier to an active biochemical defense matrix. Building that active matrix means first addressing the failure points of traditional chemical filters. The most pressing issue is that active ingredients regularly permeate the stratum corneum and enter systemic circulation. Let's look at the data on a Franz cell assay graph. The FDA has established a strict safety threshold for the systemic absorption of sunscreen active ingredients, capping it at 0.5 nanograms per milliliter. Clinical testing data for traditional filters reveals a significant problem. Chemicals like Avobenzone breach this threshold within a single hour. Oxybenzone, a common filter, has spiked to 258.1 nanograms per milliliter, eclipsing the safety limit by orders of magnitude. In tropical climates, where high humidity and heavy daily reapplication are mandatory, this level of absorption increases the risks of dermal sensitization and long-term toxicity. The structural solution to this permeability is atomic scale containment. Next-generation formulations entrap UV absorbers, like Avobenzone and octocrylene, inside sol-gel silica glass microcapsules. By utilizing polysilicon 14 microencapsulation, the active compounds are surrounded by a microscopic protective shell. This prevents the chemical filters from making direct contact with the epidermis. Returning to the Franz cell assay graph, we can see the clinical validation of this encapsulation. The new formulation produces a perfectly flat line, maintaining 0% penetration for the first 6 hours of wear. This atomic scale engineering isolates the protective chemistry from the body. It allows formulators to use high-concentration chemical filters to achieve intense UV blocking without compromising systemic biosafety. Mineral filters, like titanium dioxide, naturally solve the absorption problem because they sit securely on top of the skin. However, they introduce severe optical limitations. Traditional mineral sunscreens rely on zero-dimensional rutile titanium dioxide nanoparticles. These dense, spherical structures scatter incoming visible light in all directions, creating an opaque white cast that is highly problematic for darker skin tones. To eliminate the white cast, scientists have modified titanium dioxide into a two-dimensional nanostructure. This structural reconfiguration physically alters the optical properties of the mineral. The new material features a micro-sized lateral dimension of 1.6 micrometers, but it is flattened to an atomic scale thickness of just 1.2 nanometers. At this thickness, it stops deflecting visible light and instead lets those waves pass straight through. The optical data confirms the success of this geometry. The 2D structure achieves over 80% visible light transmittance and reaches a natural appearance factor of 0.99 out of a possible 1.0. Beyond the optical clarity, this layered architecture offers a secondary biochemical benefit. The 2D sheets suppress the generation of reactive oxygen species by a factor of 50 compared to traditional nanoparticles. By engineering the mineral down to a 1.2 nanometer thickness, formulators have successfully decoupled high-performance UV blocking from the cosmetic penalty of a white cast. Even with microencapsulated chemical filters and ultra-thin mineral sheets, we still have to account for that original 2% UV leakage. To manage this, we have to look inside the nucleus of a human keratinocyte. When residual UV radiation penetrates a cell, it targets the genetic code. The energy causes adjacent thymine bases in the DNA to unnaturally fuse together. This forms structural defects called cyclobutane pyrimidine dimers, creating physical kinks in the DNA helix. To handle these lesions, modern formulations deploy an active biochemical defense, liposome-encapsulated DNA repair enzymes, specifically targeting these mutations using an enzyme called Photolyase. Photolyase is an ancient repair mechanism produced by almost all living organisms on Earth to survive daily sun exposure. However, placental mammals, including humans, completely lack the ability to produce it. By applying it topically, we can utilize its exact biological function. The Photolyase enzyme features a specific binding pocket that recognizes and docks onto the warped geometry of the DNA lesion. Once attached, the enzyme initiates a catalytic process called photoreactivation. It absorbs ambient blue light wavelengths to power a chemical reaction that breaks the mutant bond. The DNA helix snaps back to its healthy, original form. Phase 3 clinical trials demonstrate the efficacy of this process. Subjects prone to skin cancer who applied these liposome-encapsulated enzymes developed significantly fewer precancerous and cancerous lesions over a 1-year period compared to a placebo group. Incorporating these enzymes adds an active layer of cellular repair to the formulation, reversing UV-induced mutations in real time. All of this microscopic engineering fails if the product slides off your face. In extreme tropical microclimates, constant perspiration and high ambient humidity systematically dismantle standard SPF films. SWAT compromises protection through 2 distinct mechanisms. First is direct wash-off, which physically thins the barrier. Second is spatial redistribution, where moisture forces the film to pull apart, leaving dangerous gaps in your coverage. The baseline defense against moisture relies on building a matrix of hydrophobic film formers, like polysilicone 14, combined with very small water-absorbing particles that resist physical breakdown. But formulators eventually hit a physical limit. Increasing the concentration of heavy film-forming agents makes the formula too thick. This negatively affects how evenly it distributes over the skin, resulting in a heavy, unbreathable texture. To solve this, researchers developed dynamic film technology. They integrated specific soap-derived ingredients designed to actively react with metal ions, like calcium and magnesium, that are natively present in human sweat. When perspiration hits this formulation, an ionic reaction occurs. The moisture triggers the film to instantly tighten its structure, dynamically increasing its water repellency and adhesion to meet the strict FDA 80-minute water resistance standard. Modern formulations no longer treat sweat as a threat to film integrity. Instead, they use perspiration as a chemical catalyst to actively fortify the barrier precisely when it is under the most environmental stress. The performance of a modern SPF is defined by its direct interaction with cellular biology. Look at the macro-level defense. 2-dimensional titanium sheets and encapsulated chemical filters block broad-spectrum radiation without leaving a white cast, sitting securely on the stratum corneum, ensuring zero systemic absorption into the bloodstream. Simultaneously, the ionic polymer matrix fortifies the surface against humidity. When that inevitable 2% of UV slips through, Photolyase enzymes inside the nucleus actively repair the genetic damage. This layered matrix specifically targets the historical gaps that cause hyperpigmentation, barrier degradation, and photoaging in high UV tropical environments. By combining precise nanoscale material science with exogenous biochemical repair, the modern SPF abandons the concept of an incomplete passive shield. It acts as an active, resilient extension of your skin's own cellular homeostasis.